Hey everyone, welcome to Introduction to Cognitive Science. I'm Adam, and the focus of our lecture today is on brain-computer interfaces, or BCIs. The article that we read for today was entitled Brain-Computer Interfaces in Neurological Rehabilitation, published in the journal The Lancet Neurology in 2008 by Daly and Wolpaw. Today, we will focus on brain-computer interfaces, or BCIs. So let's begin by reviewing some basics about neural activity, as well as invasive and non-invasive techniques for recording neural activity. We will then discuss how recorded neural activity, along with relevant training procedures, can be used in brain-computer interfaces to help individuals restore functionality for daily living, including the ability to communicate with others on a computer and the ability to move prosthetic limbs. Since BCIs make use of neural activity to determine relevant actions to be performed, let's begin by reviewing neural activity and the different frequency bands that neurons produce during perception, cognition, and action. The brain generates neural activity that is used in brain-computer interfaces. Neural signals can be measured in terms of spikes or in terms of local field potentials, or LFPs. Spikes reflect the action potentials of individual neurons and are therefore acquired primarily through microelectrodes implanted with invasive techniques. Local field potentials, on the other hand, are measures of combined synaptic, neuronal, and axonal activity of groups of neurons and can therefore be measured by EEG or by implanted electrodes if greater spatial resolution is required. Most BCI research currently uses signals from EEG or electroencephalography. Let's look at an example where EEG is recorded from 64 channels on the scalp during an auditory entrainment task. In this auditory entrainment task, a speech sound such as da is produced at a regular and steady rate, while participants passively listen to the sound. As participants passively listen to the regular and steady sound, rhythmic oscillatory activity is entrained to the repetition rate of the sounds. We then take the EEG waveforms from each channel and perform a high-resolution spectral temporal analysis that separates the signal into a series of frequency-specific bands that can be represented in a three-dimensional time-frequency plane. Here in the figure, top left, we see a picture of someone's head and the positioning of the EEG for recording. Okay, so this is just standard positions for on the scalp to record EEG data. Here we see that this is the EEG signal. Here we see that a time frequency analysis is performed on the EEG signal. Next, we have a principal component analysis that is performed, which gives us this. Then we have an independent component analysis, which is performed, which gives us this. And then finally, we have a current density reconstruction, which provides us with this. During this auditory entrainment task, information enters the nervous system at lower regions, while higher level processing takes place at higher regions of this axial-spatial hierarchy. Okay, so when you're engaged in an auditory entrainment task or any sort of task, right, perceptually based task, there's going to be lower sensory level information that occurs at a lower level. And then as we progress through the axial-spatial hierarchy, it will be processed at a higher and higher level. Okay, so on the right-hand side, we see processes that correlate with the axial-spatial hierarchy. So at the lowest level, we have transduction, then we have encoding, selection, perception, cognition, and then action. Okay, and this is correlated with information processing occurring up through the axial-spatial hierarchy. We also notice that there's a spectral hierarchy that's associated with the axial-spatial hierarchy. Okay, neural circuits at the lower level, so neural circuits at the lower level here generate oscillatory activity that is relatively higher in frequency. And we see that here, right? Neural circuits at the lower level generate oscillatory activity at a higher frequency, whereas neural circuits at a higher level generate oscillatory activity at a lower frequency. When detecting a stimulus, peak changes in high frequency oscillatory activity occur earlier than peak changes in low frequency oscillatory activity. And this is just another slide to help reinforce this point, right? Peak changes in high frequency oscillatory activity occur earlier or first. And that just means peak changes at the lower level are going to occur first or earlier than peak changes at a higher level. Okay. And here we can see that, right? We're going to have a lower level processing here at the level of auditory brainstem response. We'll have higher processing here, middle latency and cortical responses, and then even higher levels of processing here with late ERPs or late event related potentials. Okay. So we see at the lowest level, so in terms of like bottom up processing, here are the stages from the bottom to the top. Okay, when we're engaged in this sort of auditory entrainment task, right? We're going to get stimulation at the lowest level first, and then it's going to progress upward through the axial spatial hierarchy, right? So just to help you understand this bottom up processing, first we're going to get activity at the auditory nerve, right? Cranial nerve 8, right? And we see that here, cranial nerve 8, right? Then we have the cochlear nucleus or cochlear nucleus, then the superior olivary complex, then the nucleus lateral lemniscus, and then the inferior colliculus, okay? Progressing up from the bottom, the auditory nerve or cranial nerve 8, up to the inferior colliculus here in terms of auditory brainstem response, okay? And we see here that there's the oscillatory activities at a higher frequency at this lower circuit level. Okay. Next, information is going to move up to the medial geniculate body, right? Then the primary auditory cortex or A1, and then up to secondary auditory cortex and auditory association cortex or A2 and AA. Okay. Notice here at a higher level of neural circuitry that we have lower frequency oscillatory activity. Okay, and then finally, we progress up to cognitive areas and areas involved in executive control. Okay, and here we see that at the highest level, in terms of neural circuits, we get the lowest frequency of oscillatory activity. Okay, and then peak changes in this activity are going to occur with the highest frequency or the lowest level first, and then last will be the lowest frequency or highest level of neural circuits for processing this information. Okay, so in other words, this is bottom-up processing from the bottom up to the top. And here, in addition to a description of bottom-up processing with auditory information, I've also flagged for you or identified for you some important responses, types of response, right? We have auditory brainstem responses, right? Responses 1, 2, 3, 4, and 5. We also have middle latency and cortical responses, including the middle latency response, or MLR, P1, N1, P2, and N2. And then for late ERPs, right, we have P3, N4, and P6 as responses. All right. Now that we're talking about oscillatory activity, it's important that we understand some of these main concepts, okay? So what is frequency? Frequency is the number of occurrences of a repeating event per unit of time, okay? So we saw that this oscillatory activity is higher frequency than this. There's more events occurring per unit of time here than here. Okay, you can like count the peaks here, and there's more here than here. So this is a higher frequency than this. Polarity is the direction and amplitude of deflection. Okay, so like this, positive, okay, or negative polarity. Amplitude here is the maximum displacement or distance moved by a point on a wave measured from its equilibrium position. Latency is the time delay where an approximate peak voltage deflection occurs. So here, latency. And then finally, period is the time taken for one complete oscillation. All right, now let's look at the different frequency signal bands in the EEG signal. And here I've provided the different frequency bands for you. And this corresponds to activity here in the CDR spectrum, and also here in terms of activity in the brain. Okay, so in this figure, we see activity across all of the frequency bands. Here, this is delta. So each band has 2 bars. Okay, so this is the delta band here. This is the theta band here. This is the alpha band here. This is the beta 1 band. This is the beta 2 band, the gamma 1 band, and the gamma 2 band. And down here is just a visualization with a human's face, so you can understand the positioning of the head in this figure up top. Okay, so what do we see? This figure up at the top left is the left view of the brain. Okay, so if you're looking at me from the left side, okay, the top middle is a top view of the brain. Okay, so looking down on the brain. And so this is the whole left side of the brain. Here, this is the left side of the brain. This is the right side of the brain. And then over here, this is just a right view of the brain. Okay, here, down here, we get a view of the brain at a slight angle. So it's mostly the left side, but facing in a slightly backward direction. Okay, and then here is, you can see the right side of the brain here. Okay. So right side is here, left side is there, okay? And then here, this bottom middle is if we turn around, right? That's why this is sort of flipped. This is a front view of the brain. So a front view, the left is on the right and the right is on the left, okay? So if someone is facing towards you, right, our left sides are opposite to each other. Okay, so this is just how we can visualize activation in the brain. Okay, and we'll notice that the colors match from the CDR spectrum with this visualization of the brain here. Okay, so we see that there's some, actually this will be clear in the next slide. I've separated the bands so that on each slide we can focus on specific band, okay? So the important thing on this first slide is we have a nice picture of how the different frequency bands are involved in different regions of the brain. The color of activity in the brain corresponds to the color of the bars in the current density reconstruction spectrum here. The height of each bar in the CDR spectrum represents the relative contribution of that frequency at a given point in time. Okay, so at this point in time, we have a large contribution from the theta band, right? And we can see that here. This is sort of the highest bar. There's a great contribution from theta band here, okay, and relatively less from like the beta bands. All right, so let's go ahead and look at each frequency band in turn. Okay, so the delta band is a frequency. This delta band ranges from 1 to 3 hertz. Okay, and we see here, this is the delta band, and this is areas of the brain that are active, right, for this delta band. The delta band activity occurs in the midbrain limbic structures, including the thalamus and the temporal cortex. We also have the theta band. I didn't have a clear image of just the theta band, but the theta band would be here, for example, these two, right? This is the delta, this is the theta, these two, right? And we see in terms of color, this is sort of like, this is maroon, delta, theta is more red, right? Whereas alpha is orange. So if we're looking for red, we can see that theta band activity occurs here, okay? Right around there. Alpha band activity, which is next, right, is in the orange. Alpha band is from 7 to 12 hertz. And we see that alpha band activity occurs in the cingulate cortex, precuneus, and auditory cortex. The beta 1 band is from 12 to 20 hertz. And we see the beta 1 band right here. This is like a yellow and a light green. And we can see the yellow and the lime green where that's active. Okay, and that reflects beta 1 band activity from 12 to 20 hertz range. Here we also see a little bit of beta 2 band activity, and this would be 20 to 30 hertz range. Okay, and this is like a darker green, so we can see areas of darker green as well. Next, we have gamma band activity. Gamma 1 band is from 30 to 50 hertz, and the gamma 2 band is from 50 to 80 hertz. Okay, and here we see the gamma band activities in blue. Okay, and so we see in the blue that this activity occurs in the cingulate cortex, temporal cortex, auditory cortex, and even in the cerebellum. All right, so again, we have different frequency bands, and the delta band occurs from 1 to 3 hertz. The theta band occurs from 3 to 7 hertz. The alpha band occurs from 7 to 12 hertz. The beta 1 band occurs from 12 to 20 hertz. The beta 2 band occurs from 20 to 30 hertz. The gamma 1 band occurs from 30 to 50 hertz, and the gamma 2 band occurs from 50 to 80 hertz. Okay. And this processing occurs in different areas of the brain. So, electrophysiological components of particular interest for brain-computer interfaces include oscillatory EEG activity, event-related potentials, slow cortical potentials, and neuronal potentials. Oscillatory EEG activity is caused by complex networks of neurons that create feedback loops. This synchronized firing of the neurons in these feedback loops generate observable oscillations. The frequency of oscillations decreases as the number of synchronized neural bodies increases. Okay, and that's why we see as we move up the axial spatial hierarchy, right, we're incorporating more components, that the frequency of oscillation decreases, right, because the number of synchronized neural bodies increases. Here in this figure, we see event-related desynchronization, or ERD, and event-related synchronization, or ERS, before and after a movement onset. ERD, or event-related desynchronization, is the result of a decrease in the synchronization of neurons, which causes a decrease of power in specific frequency bands and can be identified by a decrease in signal amplitude. ERS, or event-related synchronization, is the result of an increase in synchronization of neurons, which causes an increase of power in specific frequency bands and can be identified by the increase in signal amplitude. Right? And we see here ERD, event-related desynchronization, right? This is a decrease in the synchronization of neurons, which is also reflected by a decrease in signal amplitude. And we see that here, right? It's sort of a smoothing out right here in the ERD, whereas here in the ERS, event-related synchronization, that here we have increased synchronization of neurons, which causes an increase in amplitude, right? It's the opposite of a smoothing effect here, right? We're creating some spiking activity here. And here on this figure, we have the techniques required to extract event-related desynchronization and event-related synchronization from a raw EEG signal. So we have a raw EEG signal from each trial, which is band pass filtered. The amplitude samples are then squared to obtain the power samples. The power samples are then averaged across all trials. And then variability is reduced and the graph is smoothed by averaging over time samples. Okay, and we see that in each case. Okay. Many BCI systems utilize components in the alpha and beta bands between 7 and 30 hertz. Right? And we see that here. Here's 8 to 11 hertz, and here's 26 to 30 hertz. Okay, utilizing alpha and beta bands. All right, ERPs, or event-related potentials, are also an important component. One very popular ERP component is the P300, which is considered the oddball paradigm. Okay, this is the P300 is a component that we see in the oddball paradigm. Okay, and what is an oddball paradigm? Well, I'll describe it for you right here. So if you're randomly presented with different objects on a screen, excuse me. So imagine I'm providing you with different objects, like random objects, but I want you to look for one in particular. Okay. Well, the P300 component occurs when that object that you're looking for is finally flashed, while this component won't occur when you're presented with the other examples of stimuli. Okay, maybe I'm presenting you with different pictures and I'm telling you to identify the car or the dog, right? You'll have a lot of random images, but when you're finally presented with the target image, right, that will elicit this P300 component. Okay. This is in some sense the odd one out, the oddball, right? That one picture of the dog stands out as the oddball against the rest of the random stimuli. Okay. Also, the amplitude of the P300 is inversely proportional to the rate at which the object of interest is presented, right? You're going to have a greater response to the extent that it's a greater oddball, right? You're going to get a wicked P300 if like there's like no pictures of dogs, and then finally there's that one, right? You're going to get a nice P300 response, which is inversely proportional to the rate at which the object of interest is presented. Okay, and this occurs approximately 300 seconds after the object is displayed. And although we call this a P300 because it occurs approximately 300 milliseconds after the object is displayed, in reality, this response is elicited somewhere between like 250 milliseconds to like 500 milliseconds after stimulus presentation. Okay, so P300, but we might see that the P300 is happening like closer to 400 or even like 250. Okay. The P300 is a natural response and requires no training. Here's how we can read different results from event-related potentials. Okay, we'll see that event-related potentials are indicated using a letter and a number, right? Like P300 or N300 or P400 or P600 and N400, right? These are different responses. Okay, and the way that we read this is the P or N is going to indicate either a positive for P or a negative for N deflection. Okay, so if it's a P300, that means there's a positive deflection that occurs at 300 milliseconds after stimulus presentation. Okay, an N300 means that there's a negative deflection that occurs at 300 milliseconds after stimulus presentation. Okay, we also have an N400, and quiz yourself, what does that mean? Well, we have a negative deflection at 400 milliseconds approximately. And the P600, what does that mean? Well, it means we have a positive deflection at 600 milliseconds. Okay, and these are indicative of different aspects of cognitive processing. Okay, and as you read these responses on the figure, just make sure that you're familiarize yourself with which way is positive and which way is negative. Okay, so here we have an example of a P300, right, where a positive deflection is going to go down, right? We see that here's 0, but then we get 50 and 100, 150, and then going downward and going up, we have negative, right? Like negative 50, negative 100, right? So we see here that we get a positive deflection around 300 milliseconds after stimulus presentation. Okay, and that reflects our desired choice, right? That thing you were looking for, the image of the dog, as opposed to all the other random choices. Okay, so that's how we can read this event-related potential. Okay, and we should all be familiar with this now, since we've been working in this course for about a month now. We know that the x-axis here, right, this represents time in milliseconds, right, and the y-axis represents voltage. All right, here we see that different slow cortical potential signals also convey different information about intentions for action. Slow cortical potentials are caused by shifts in the depolarization level of certain dendrites. Slow cortical potentials occur from half a second to 10 seconds after the onset of an internal event, and are therefore considered slow cortical potentials. Okay, here we're looking at somewhere between half a second to 10 seconds, right? So 10 seconds, that's a very slow cortical potential, slow cortical potential, SCP. Okay. And then finally on this figure, we see neural potential signals, which are voltage spikes that can be measured from individual neurons or groups of individual neurons. And here we have a signal from an idling neuron. And then down here, we have a signal from an active neuron. Okay, we see there's more activity here than up here. All right, so neural activity for BCIs can be recorded using invasive and non-invasive techniques. Non-invasive techniques such as electroencephalography or EEG have relative strengths and weaknesses. Okay, we see that EEG is going to be measured on the scalp. Okay, and some of the strengths of this approach is that it's easier to use and safer to use than ECOG and implanted electrodes. Okay. EEG also has weaknesses though, right? Or limitations. And that includes limited resolution and frequency range compared to ECOG and implanted electrodes. Okay, so we see that being recording signals at a more superficial level, right? On the outside of the head, that this is easier and safer. However, we're gonna not have the fine grain resolution as if we, for example, had an implanted electrode and we're reading from like a single neuron, right? So we see that there's a trade-off here. We also have invasive techniques for recording neural activity, such as ECOG and recordings from specific neurons using implanted electrodes. But invasive techniques also have their relative strengths and weaknesses. Strengths of ECOG include higher resolution than EEG and easier than and safer than implanted electrodes. Weaknesses of ECOG include that it's riskier than EEG, but while also having lower resolution than implanted electrodes, right? So by holding on to this sort of intermediate position here, we see that its strengths are that it's safer than what's down here, right? But it's of lower resolution than what's up here. Okay. And then finally, we have implanted electrodes, right? We can implant electrodes all the way down in the brain. And strengths is that this is going to provide the highest resolution and frequency range compared to ECOG and EEG, right? But clearly, there's risks to this approach as well, like safety risks, okay? So, strengths and weaknesses or strengths and limitations for each approach, okay? And it's important to consider this as we think about the particular individual that we want to assist with BCIs. All right, so let's go ahead and look at an invasive technique. This figure shows how to record brain activity using invasive signal acquisition methods. So here, a cone-shaped glass electrode is implanted through the skull and directly into specific neurons in the brain. That's what we mean by invasive, right? We're going to put this into the skull. This electrode, right, is filled with a neurotrophic factor that causes dendrites to grow into the cone and contact one of the gold wires that transmits the electrical activity to the receiver unit outside of the head, which is then amplified and transmitted to the BCI controller using a transmitter. Okay, and we can get a nice picture here. Okay. So here we see a coaxial optrode for simultaneous electrical recording and optogenetic neuromodulation. On the top here, we have a schematic of the coaxial optrode, right? So this is just like a schematic diagram, right? And we see that this is the optical fiber, this is the stainless steel tube, right? And here's the gold coating, okay? So that's what's provided in A as a schematic. In B, we have an image of the coaxial optrode tip from a scanning electron microscope. Okay, so it's cool that we can see this, right? And notice how small this is, right? This is measured in micrometers. Okay. All right, and then so yeah, here we have the image from the electron microscope here. Okay. And here we see that this is 20 micrometers, this unit. Okay. In C, we have a full-length photograph of the coaxial optrode here. Okay, and here's the wire, optrode tip. D is a photograph of the coaxial optrode depicting reinforcing thin stainless steel tubes and tissue. All right, so here's the stainless steel tube and the optical fiber in the tube. And then in this figure, we have performance of the coaxial optrode. We see the performance of the coaxial optrode in vivo electrophysiological recordings from the somatosensory cortices of a mouse, rat, and non-human primate. Okay, so this is an anesthetized mouse, an anesthetized rat, and an awake behaving non-human primate. Okay, the blue and green bars indicate timeframes of light delivery, highlighting light-induced increases in neuronal spiking activity. Okay. Cool. So this here, what's nice about this image is we just get a picture of what's going on in this drawing, okay, this depiction here. Okay. So individual electrodes in this Utah electrode, this is called a Utah electrode, are tapered to a tip, penetrating only 1 to 2 millimeters into the brain. Research in animals such as rats and monkeys has led to the rapid development of microelectronics that enables recording of electrophysiological activity from a small group of neurons or even a single neuron. Although there are benefits to using invasive techniques, such as increased spatial resolution, they can also cause significant discomfort and risk to the patient. Therefore, researchers use invasive techniques in humans only if it will provide considerable improvement in functionality over available non-invasive methods. So another approach that is less invasive than implanting electrodes, right, directly into the brain, and that has been well applied to epileptic patients for surgical planning, is patching subdural electrode arrays over cortex to record electrocardiogram signals or ECOG signals. So here, right, we see that EEG is positioned on top of the scalp, right? You can just wear an EEG, a piece of EEG equipment on the outside of the head. With ECOG, right, we see that we're getting recordings here, okay? This is where we're getting recordings from electrocardiogram signals, okay? The implanted electrodes, right, are here, even deeper, okay? See, they're penetrating into the cortex, okay? But with ECOG or electrocardiogram signals, we have subdural electrodes, right? So subdural electrodes here, which are closer to the neuronal structures in superficial cortical layers than EEG, right? Compared to here, right? Our ECOG is able to record signals closer than EEG is. The advantages of subdural recordings over scalp recordings include recording from smaller sources of synchronized activity, higher signal to noise ratio, and improved ability to record gamma activity above 30 hertz. Remember that the higher frequency activity is going to occur lower in lower neural circuits. Okay, so by getting deeper in there, we can have an improved ability to record that higher frequency activity, such as gamma activity above 30 hertz. Gamma activity is generated by rapidly oscillating cell assemblies composed of a small number of neurons. Consequently, gamma activity is characterized by small amplitude fluctuations that are not easily recorded with scalp electrodes using EEG. Levine and colleagues implemented a direct brain interface that takes voluntary commands directly from ECOG signals in epileptic patients. The cortical locations of the subdural electrodes were based on clinical considerations relating to epilepsy surgery. The subjects were instructed to make different movements of the face, tongue, hand, and foot in a prompt-paced or self-paced manner. Half of the ECOG recording was used to produce an average ECOG segment and create ERP, or event-related potential, templates. Then the cross-correlation of templates with the continuous ECOG signal was used to detect ERPs that correspond to specific movements. The accuracy of ERP detection for the 5 best subjects was over 90%. In another experiment of self-paced movement using ECOG, it was reported that self-paced movement was accompanied by a relatively widespread alpha and beta ERD, as well as a more focused gamma ERS in the 60 to 90 hertz frequency band. And remember that ERD stands for event-related desynchronization, and ERS stands for event-related synchronization. All right, so what processes are required to convert the user's intent from a raw EEG signal into device action? Well, now that we have, that we know how to identify neural signals in the brain, BCIs need to make use of these signals. So BCIs extract features from signals and translate them into logical control commands. A feature in the signal is a reflection of a specific aspect of the physiology and anatomy of the nervous system. So the goal of feature extraction is to identify a physiological aspect of the nervous system across a specific time series. The steps involved in feature extraction and translation are highlighted in this figure below. Signals are captured through invasive or non-invasive methods. Since signals contain noise, feature extraction and translation are applied to remove that noise. There are various feature extraction techniques that can be performed, but the general focus is on maximizing the signal to noise ratio. And feature translation techniques are then used to classify the relevant features into one of the possible states for a decision. All right. Next, we need to understand the important role of training to get BCIs to work. So when we first learn to move our arms and talk to others, a period of practice and refinement is usually required. Similarly, the use of BCIs usually requires a training period. Early research on BCIs used animal models. For example, in this earlier work, Nicolelis and Chapin were able to have monkeys control a robot arm in real time using electrical discharges that were recorded with microwires that were implanted beside a single motor neuron. Various motor control parameters, including the direction of hand movement, grip force, hand velocity, and acceleration, were derived from the parallel streams of neural activity using mathematical models. In this system, monkeys learned to produce complex hand movements in response to arbitrary sensory cues. The monkeys could then exploit visual feedback to determine how well the robotic arm reflected their hand movements. This design was used to test a closed loop control brain-machine interface for motor control in the macaque monkey. Chronically implanted microwire arrays were used to sample the extracellular activity of populations of neurons in several cortical motor regions. Linear and nonlinear real-time models are used to extract various motor control signals from raw brain activity. The outputs of these models are used to control the movements of a robotic arm. For instance, while one model might provide a velocity signal to move the robot arm, another model running in parallel might extract a force signal that can be used to allow a robot gripper to hold an object during an arm movement. Artificial visual and tactile feedback signals can be used to inform the animal about the performance of a robot arm controlled by brain-derived signals. Visual feedback can be provided with a moving cursor on the video screen to inform the animal about the position of the robotic arm in space. Artificial tactile and proprioceptive feedback can be delivered by a series of small vibromechanical elements attached to the animal's arm. This haptic display is used to inform the animal about the performance of the robot arm gripper, whether the gripper has encountered an object in space or whether the gripper is applying enough force to hold a particular object. So we see that early work has focused on non-human primates. Okay, this is generally the case in psychological research. The effectiveness of BCI is dependent on the capacity of the user to willingly and consistently control their EEG activity. Unlike motor tasks, control of brain activity is harder to achieve since the user can neither identify nor discern the activity. The user can only comprehend their EEG activity through the feedback received from the components in the BCI system. Importantly, the goal of training is to have users voluntarily produce detectable EEG signals that can be altered to achieve a specific result. Although the user might not be aware of how and when the signals are generated, the signal generation process often involves voluntary brain activity performed by the user. BCI systems differ in whether these voluntary signals must be produced through conscious mental activity, such as by adding numbers, or a more automatic response to the situation that requires minimal conscious effort. And here is a figure of an idealized bidirectional brain-computer interface for closed-loop prosthetic control. Here, neural correlates of motor intent are recorded from electrode arrays implanted in motor areas of the brain, such as primary motor cortex. Okay, so I might be trained on a motor task where they want me to intend to perform a motor action, in which case they're looking at the EEG signal that's produced from that motor intent, right? Then we can take that EEG signal, right, and use it for, we can use it for the prosthetic arm. So we're gonna record that motor intent, right, using electrode arrays implanted in motor areas such as M1 or primary motor cortex, right? Those signals are then decoded and used to control the movement of an awesome prosthetic arm, such as the arm right here. Okay, maybe I don't have functionality of my arm anymore. Maybe I was in a car accident or in some sort of accident, and I no longer have the ability to move my arms, right? So what I can do is through training of motor intent, we can get the signal that I'm consistently producing and then use that signal, right, to control the movement of this prosthetic arm. Sensors on the robotic arm detect info about touch via contacts with external objects or proprioception via movement and position of the prosthetic arm. Outputs from these sensors are then converted to patterns of stimulus pulses that are delivered via implanted electroarrays to sensory regions of the brain, such as primary somatosensory cortex. Okay, so what's going on here is I can be trained to produce some EEG activity, like by having some motor intent. Okay, that intent is recorded from primary motor cortex. Okay, those signals are then decoded to move this prosthetic arm. The movement of the prosthetic arm then provides feedback, right? As the arm begins to move, sensors from that arm are then converted into stimulus pulses that are then communicated back into somatosensory cortex, primary somatosensory cortex. Okay, and you see now we have a loop going on here where my motor intent is generating signals that are used to move the arm, but the movement of the arm is then generating signals back to somatosensory cortex, giving me feedback of that movement. Okay, and then based on my feedback, I can then modify my motor intent, right? Which is going to modify the signal I produce, which is going to modify the movement of the arm. And that modified movement of the arm is going to send a modified signal back to primary somatosensory cortex. And I can continue doing this loop. Okay, so as I reach for this drink because I'm thirsty, right, I'm sending a motor command, right, which is being read by the prosthetic arm. And then once I reach my drink, I'm going to get feedback that I've reached it, right, which is given back to primary somatosensory cortex. And now that I know I've reached my destination and I have a nice grip of my drink, then I can now extinguish the reaching behavior. I can now adjust my motor intent, okay, which then adjusts the signal for the arm and so on, okay? Essentially the same way that we learn to use our hands for reaching. In a similar way, we learn to use prosthetic devices for reaching also. Okay, one of the major differences is there's going to be like a bandwidth difference here. Whereas when I'm interacting with all my components normally, right, it's happening very quickly and seamlessly, right? The loop here is closed and very quick, right? I have like very quick, continuous online feedback from my reaching behavior, right? This whole loop is closed and happening very quickly, whereas with prosthetic devices, it's going to be slower, okay, and less fluid. But as technology advances, hopefully we can close this loop and make this happen in something like real time, okay? And it raises interesting questions, such as at what point will we be able to move prosthetic limbs with the same speed and fluidity as we move our real limbs? Right? And if this is the case, will this enable us to have the ability, for example, to conduct surgery over long distances, right? Maybe I'm like the best neurosurgeon in the world, but I don't need to travel anymore to different countries to perform surgery, but I can just sort of, right, using recordings from motor cortex and feedback, right, I can maybe perform a surgery in South Korea or Australia, right? And almost like feel as if I'm using my real arms in performing these surgeries. Okay, so this is the idea here about an idealized bidirectional brain-computer interface for a closed loop prosthetic control. So most training methods require the user to perform some specific cognitive task, right? Notice here that we need to train someone, the participant, to generate signals, like a motor signal here, which can be recorded and transmitted to the prosthetic device. Okay, so this is going to require some training. All right, and there's different ways that we might train such a device. Some common ways include arithmetic training, motor imagery training, and these methods focus on developing the user's ability to generate EEG components through voluntary and conscious mental activity. So for example, a motor imagery task, which is among the most widely used, in this sort of task, users imagine several motor movements, right? Such as moving the left or right hand. Research has shown that this generates signals from the sensory-motor cortex of the brain and can be identified with EEG. After several training sessions, the user is able to control the amplitude and frequency of the required electrophysiological component. Okay, so for these participants, I'll have them perform different motor imagery tasks, like imagine moving your left arm or your right arm. Okay, and as they continue to practice this imagining task, they're going to become increasingly better at generating a consistent EEG response, right? And that consistent EEG response can now be used as a signal to move the arm one way as opposed to the other. Nairhaus and colleagues in a recent paper found rapidly and spatially specific signs of brain plasticity measured with structural MRI and functional MRI after only a single hour of mental BCI training in BCI naive subjects. Okay, so participants that had never used BCI before, after a single hour of training on the BCIs, right, they found brain plasticity, evidence of brain plasticity. Okay, Nairhaus and colleagues employed two BCI approaches with neurofeedback based on modulations of EEG rhythms by motor imagery and event-related potentials elicited by visually targeting flashing letters. So they used two different types of tasks here. Motor imagery task and identifying visually targeting flashing letters. Okay. Nairhaus and colleagues found an increased signal in the occipital and parietal areas after training on motor imagery. They also found an increased signal in the precuneus and sensory motor regions after training on visually targeting flashing letters. Their analysis showed that brain plastic changes are spatially specific for the respective neurofeedback. Okay, so importantly here, we see a couple of things. One is that in a short amount of time, individuals are able to train on these different components, right? They're able to train on motor imagery as well as on visually targeting flashing letters, two different types of tasks, right? And they're able to gain control in these areas relatively quickly. Okay, so one important finding is sort of the quickness of the training. Another has to do with sort of the discrimination in the training, right? In that the motor imagery training and the training on visually targeting flashing letters recruit different areas of the brain. Okay, so different types of cognitive tasks rely on different brain areas. And this is really awesome and helpful to know because uh, one of the reasons, recall, that people have BCIs is because they suffer some sort of brain damage or injury, right? So it may be the case that a particular individual has has damage to a particular area of the brain, right? So maybe the visually targeting of flashing letters, maybe that's not a great task for a specific individual to perform because they have lesions in that area, or there's degraded connectivity or functionality in that area. Okay, well, what this study is showing us is that, well, motor imagery is going to occur in a different area, right? So we can then train those individuals or that individual with this type of task, a motor imagery task. So even if areas of the brain responsible for visually targeting flashing letters, even if that area is non-functional or not healthy, we can train these individuals on motor imagery tasks. And as long as they're able to improve on this motor imagery task, where they can produce a consistent EEG signal, we can use that signal, right, for our prosthetic limb or for our BCI device. Okay, and we can also get this going relatively quickly. The results from this study suggest that after 1 hour of training, right, we get some nice results. All right, I had I had a phone call, so I had to pause real quick. All right, other commonly used cognitive tasks include mental arithmetic and geometric figure rotation. Okay, so there's many different tasks that can be used for BCI training. Research has shown that these tasks produce discernible components detectable by EEG. Again, the goal of training is to have users voluntarily produce detectable EEG signals that can be used to achieve a specific result, such as movement or communication. Another approach in BCI research is the operant conditioning approach, which does not require the user to perform specific cognitive tasks. The focus of this method is on helping the user gain automatic control of the device. The feedback provided by the system serves to condition the user to continue to produce and control the EEG components that have achieved a desired outcome. With continuous practice, the user is able to gain control of the device without necessarily being aware of the specific EEG components being produced. Operant conditioning methods often use motor imagery tasks to initially acclimate users to the concept that brain waves can be controlled. All right, now let's consider some factors that affect training. Both methods of training, both cognitive tasks and operant conditioning, are subject to external factors. Some of the most common factors are distractions, frustration, emotional state, and fatigue. It is important to counteract these factors during training by providing ample feedback and varying the duration or frequency of the training sessions. The EEG components produced by cognitive tasks are also vulnerable to the amount of direction provided to the user. Motor imagery is subject to issues such as first versus third person perspective, visualization of the action versus retrieving a memory of the action performed earlier, imagination of the task as opposed to verbal narration, and so on. Research has yet to prove whether users can effectively control such fine-grained details to produce significant changes in the components that they produce. Also, some users have suffered from mentally debilitating diseases that do not allow them control of all areas of the brain. So users need to be paired with the cognitive tasks that best suit their capabilities, as I mentioned in the previous slide. It is possible to discern different cognitive tasks based on the EEG components generated when the task is achieved. It is important to choose tasks with contrasting EEG components for easy discrimination. Okay, so if we want to have an individual be able to perform different motor functions or different functions with the BCI, we want to give them different distinct tasks so that way they can generate distinct EEG patterns, okay? And the BCI is going to use these distinct patterns for distinct actions, okay? So we want the tasks that BCI participants are trained on to be distinct enough so that way the command is not, there's not too much overlap between the commands, okay? So I might have a command of motor imagery, pushing something with my left arm or kicking something with my right foot, okay? And these would be different. It would help me produce a cleaner, more distinct set of signals for distinct actions. Whereas if my two tasks were motor imagery of moving my left hand, my left pointer finger versus motor imagery of moving my left middle finger, we see that that's not so distinct, right? And there's going to be a lot of overlap in the EEG signal. So it's not the ideal way to produce distinct signals for distinct commands with our BCI. Okay, so we want to give our individual distinct tasks to train on, but make sure that the distinct tasks are all suitable or appropriate for our BCI user. So it's important to choose tasks with contrasting EEG components for easy discrimination by the BCI, and it's also important to maintain consistent training regimens to ensure that subjects retain their ability to control their EEG components, okay? So it's not just like we can do this once and we're done, right? We need to continuously practice and train to make sure that we retain control of our consistent EEG signal, okay? Sort of like when we sing, if you're an excellent singer, you probably have very consistent production of pitch, right? If I tell you to sing a C sharp, right? If you sing every day, if you're a professional singer, if you train every day, you're gonna be able to produce that consistent C sharp, right? Like on cue, right? On command, right? However, if you haven't trained as a singer in years, or you only train very irregularly, your production, right, is gonna be inconsistent, okay? Similarly, we're not going to be able to generate consistent EEG signals if we don't train consistently. Okay, so it's very important that we train on different tasks as well as consistently on these different tasks. So that way we retain control of consistent yet distinct EEG signals to feed to a BCI. So that way we can use that BCI to do and continue to do distinct things. Okay, like reaching for desired objects and also communicating basic forms of information over text or email. All right, so we covered quite a bit today. What did we cover today? Well, today we reviewed some basics about neural activity and the different frequency bands that neurons produce during perception, cognition, and action. Frequency signal bands in the EEG, frequency bands in the EEG signal included the following, right? We looked at delta bands, which occur from 1 to 3 hertz. We looked at theta bands, which occur from 3 to 7 hertz. We looked at alpha bands, which occur from 7 to 12 hertz. We looked at the beta 1 band, which occurs from 12 to 20 hertz. We looked at the beta 2 band, which occurs from 20 to 30 hertz. We looked at the gamma 1 band, which occurs from 30 to 50 hertz, and the gamma 2 band, which occurs from 50 to 80 hertz. we saw that the higher frequency oscillations are produced by lower level neural circuits, and the lower frequency oscillations are produced by higher level neural circuits. We also reviewed how neural activity for BCIs can be recorded using invasive and non-invasive techniques. And we also considered strengths and weaknesses of invasive and non-invasive techniques. Okay, we looked at non-invasive techniques, in particular EEG or electroencephalography, right? And we saw that strengths of EEG include that it's the safest and easiest to use, since we are going to record electrical activity from the scalp. Okay, a limitation or weakness of this approach is just limited resolution and frequency range. Okay. In addition to non-invasive techniques, we also looked at invasive techniques, right, such as ECOG and implanted electrodes. Okay, we saw that strengths of ECOG include that it provides higher resolution and higher frequency, greater frequency ranges than EEG. Okay, so the strengths of ECOG are that it doesn't have the limitations that EEG does. Okay, but the weaknesses of ECOG is it doesn't have the strengths that recording from individual neurons does. Okay, so ECOG isn't gonna have the resolution of implanted electrodes. Okay, ECOG is also riskier than EEG. And then finally, we have implanted electrodes. Strengths of this approach include high resolution and frequency range compared to ECOG and EEG. However, relative weaknesses of implanted electrodes is that since we're implanting the electrodes, right, into the cortex, we see that this is the riskiest of all the approaches, right? As we implant things into the brain, we have to worry about, right? Because when we implant things into the brain, this is an environment that is also corrosive for certain component parts, right? So these parts may have to be replaced after time. There may be some internal movement or adjustment, and any of those things are going to be harder and more difficult to fix than if this was just at a superficial level, like at the scalp. Okay, so as we think about BCIs, and as we think about what particular individuals need, right, we need to think about, weigh the strengths and limitations of these non-invasive and invasive approaches. Finally, we discussed how recording neural activity, along with relevant training procedures, can be used in brain-computer interfaces to help individuals restore functionality for daily living, including the ability to communicate with others on a computer or the ability to move prosthetic limbs. It's exciting to think about how we may be able to use BCIs to improve the functionality, health, and well-being of many different people. As technology continues to improve, it is also exciting to think about the increasing connectivity and compatibility of humans and machines. I hope you enjoyed this lecture on brain-computer interfaces, and I thank you for your attention today. Okay, have a great day, and I look forward to seeing you tomorrow for our next lecture.